Types of data in GenBank. There are a number of molecular sequences in GenBank. A fundamental distinction is that DNA, RNA based and protein sequences are stored in discrete databases. Furthermore, within each database, sequence data are represented in a variety of forms. For example, beta globin may be described at the DNA level, for example as a gene, at the RNA level as a messenger RNA or mRNA, at the protein level. Because RNA is relatively unstable, it is typically converted to complementary DNA or cDNA, and a variety of databases contain cDNA sequences corresponding to RNA transcripts. Thus, for our example of beta globin, the various forms of sequence data include the following: genomic DNA databases. Beta globin is part of a chromosome. In the case of human RBP, we will see that its gene is situated on chromosome 11. Beta globin may be a part of a large fragment of DNA such as a cosmid, bacterial artificial chromosome or BAC, yeast artificial chromosome or YAC that contain several genes. A BAC is a large segment of DNA, typically about 200,000 base pairs or 200 kilobases, that is cloned into bacteria. Similarly, YACs are used to clone large amounts of DNA into yeast. BACs and YACs are useful vectors with which to sequence large portions of genomes. Beta globin is present in databases as a gene. The gene is the functional unit of heredity. It is a DNA sequence that typically consists of regulatory regions, protein coding exons and introns. Often, human genes are 10 to 100 kb in size. Beta globin is present as a sequence tagged site (STS), that is, as a small fragment of DNA, typically 500 base pairs long, that is used to link genetic and physical maps, and which is part of a database of sequence tagged sites or DBSTS. cDNA databases corresponding to expressed genes. Beta globin is represented in databases as an expressed sequence tag or EST, that is, a cDNA sequence derived from a particular cDNA library. If one obtains a tissue such as liver, purifies RNA, then converts the RNA to the most stable form of cDNA, some of the cDNA clones contained in that cDNA are likely to encode beta globin. Expressed sequence tags or ESTs. The database of expressed sequence tags DBEST is a division of GenBank that contains sequence data and other information on single pass cDNA sequences from a number of organisms. An EST is a partial DNA sequence of a cDNA clone. All cDNA clones and thus all ESTs are derived from some specific RNA source such as human brain or rat liver. The RNA is converted into a more stable form cDNA, which may then be packaged into a cDNA library. ESTs are typically randomly selected cDNA clones that are sequenced on one strand and thus may have a relatively high sequencing error rate. ESTs are often 300 to 800 base pair in length. The earliest efforts to sequence ESTs resulted in the identification of many hundreds of genes that were novel at the time. In November 2008, GenBank had over 5 crores 80 lakhs ESTs. Currently, GenBank divides ESTs into 3 major categories: human, mouse and other. The table shows the 10 organisms from which the greatest number of ESTs has been sequenced. Assuming that there are 22,000 human genes and given that there are about 8.1 million human ESTs, there is currently an average of over 300 ESTs corresponding to each human gene. ESTs and UniGene. The goal of the UniGene or Unique Gene project is to create gene-oriented clusters by automatically partitioning ESTs into non-redundant sets. Ultimately, there should be one UniGene cluster assigned to each gene of an organism. There may be as few as one EST in a cluster, reflecting a gene that is rarely expressed, to tens of thousands of ESTs associated with a highly expressed gene. There are over 100 organisms currently represented in unigene. For human beta globin, there is only a single unigene entry. This entry currently has 2400 human ESTs that match the beta globin gene. This large number of ESTs reflects how abundantly the beta globin gene has been expressed in cDNA libraries that have been sequenced. A unigene cluster is a database entry for a gene containing a group of corresponding ESTs. There are now thought to be approximately 22,000 human genes. One might expect an equal number of unigene clusters. However, in practice there are more unigene clusters than there are genes. Currently, there are about 120,000 human unigene clusters. This figure is a schematic representation of unigene clusters. ESTs are mapped to a particular gene and to each other. The number of ESTs that constitute a unigene cluster varies from 1 to tens of thousands. On an average, there are 300 human ESTs per cluster. Genome databases at EBI. The European Bioinformatics Institute, or EBI, is an EMBL outstation located at Hingston Hall near Cambridge, UK. Since September 1994, all activities previously based at the EMBL data library in Heidelberg, Germany are located at the EBI. The database services of the EBI are the continuation and extension of EMBL data library. A central activity of the European Bioinformatics Institute (EBI) is the development and distribution of the EMBL nucleotide sequence database. The EBI also maintains and distributes the Swiss prot protein sequence database in collaboration with Amos Bayroch of the University of Geneva. Over 50 additional specialist molecular biology databases, some produced in collaboration with the EBI, are also distributed through EBI releases and network services. The first completed genomes from viruses, phages and organelles were deposited into the EMBL database in the early 1980s. Since then, molecular biology shift to obtain the complete sequence of as many genomes as possible, combined with major developments in sequencing technology, resulted in hundreds of complete genome sequences being added to the database, including archaea, bacteria and eukaryota. The EBI website is comparable to NCBI in its scope and mission, and it represents a complementary independent resource. EBI features 6 crore molecular databases, which are as follows. Number 1, EMBL Bank is the repository of DNA and RNA sequences that is complementary to GenBank and DDBJ. Number 2, Swiss prot. Number 3, TREMBL are 2 protein databases. Number 4, MSD is a protein structure database. Number 5, Ensemble is one of the 3 main genome browsers. Number 6, Array Express is one of the 2 main worldwide repositories for gene expression data, along with the gene expression omnibus at NCBI. Genome analysis of model organisms. A major application of bioinformatics is the analysis of the full genomes of organisms that have been sequenced starting in the late 1990s, including microbial genomes, the budding yeast Saccharomyces cerevisiae, the nematode worm Caenorhabditis elegans, the plant Arabidopsis thaliana, the fruit fly Drosophila and the human genome. Many additional genome sequencing projects are either being planned or are already underway. Many years of genetic and biochemical research of these model organisms, the bacterium Escherichia coli, Saccharomyces cerevisiae, Caenorhabditis elegans, Arabidopsis thaliana and Drosophila melanogaster, have led to the accumulation of a large amount of information on gene organization and function. The mouse Mus musculus is a genetic model for humans because the 2 species are so closely related through evolution. A newly identified gene in another organism can be compared to the existing database of information to find whether it has a similar function. Genes involved in human disease, for example, are sometimes found to be similar to a fruit fly gene at the protein sequence level. As a result, a similar gene or family of genes may be found in several organisms, including a model organism, but the function is not known because the gene functions have not yet been analyzed. Hence, continued biological analysis of model organisms in those areas that are not tractable by the tools of bioinformatics has many important applications.